I think we're going to go ahead and get started, so please come in and grab a seat. Thanks everybody for taking time out of busy schedules to join us today for this event. Oh, it's back. So good morning. Scott Lanyon, I'm Vice Provost and Dean of Graduate and Professional Education here, and this is the top event of the year. This is so much fun to do. I'm really glad you could join us. We're actually having 12 different colleges represented, 12 graduate students this year. That's the most we've ever had. That's colleges across the Twin Cities campus, but also representing UMD, the Swenson College of Science and Engineering. I'd like to extend a welcome in addition to all of the fans of the presenters here. I know we have several high schools here who are attending today, future graduate students. We're looking forward to seeing your applications. Also, the university's Lifelong Learning Initiative is represented here today, so thank you all for joining us. So, let me give you a little bit of background about what this is all about. The 3-minute thesis is actually something that the University of Queensland in Australia actually developed many, many years ago. We and many institutions globally have sort of picked this up, this idea up. I think there are 900 universities now that are doing a competition like this in 85 different countries. And the key here, if you're not familiar with what you're about to see, is really helping students understand how to convey complex ideas in a very short period of time without a lot of props. Right? Think of it as the 3-minute elevator opportunity where you've just run into somebody and you want to convey quickly the importance of the work that you do. This is a really important life skill regardless of the career that you pursue. So this is our 10th anniversary of doing this, a milestone, and I actually want to call out, where is Nuru? Is Nuru hiding? Oh, there you are. Nuru, can you stand up? Nuru has been sort of guiding this now for 10 years. Really appreciate all of this work. So what happens is your various colleges have been holding their own competitions in recent months, and the winner of each one of those competitions is who is here today to compete. And I don't really like to stress the idea that this is a competition. This is mostly a celebration of the science that these incredible students have been doing and are sharing with you today. Students really are building their ability to present their research succinctly and persuasively to a non-specialist audience. That's the goal here. Students are allowed to have a single slide with no audio, no animation, just a static slide, and are talking for 3 minutes or less. So, it is a competition though. So we do have judges, and I'm going to introduce them in a moment, but I want to let you know what they're looking for because I'd like you to be looking for these same criteria as you watch and listen to these presentations. Clarity and comprehension of the research, accessibility for a general audience engagement and enthusiasm, overall communication effectiveness, and importantly, and this is really hard for all of us, you're not judging the science, you're not judging the research. This is about the presentation, okay? So try to keep that in mind. So every participant today will receive a gold-level digital badge from the graduate school, sort of documenting this participation in professional development. And students who also competed at the collegiate level will receive a maroon badge. We're also awarding prizes. So the People's Choice Award, I'll talk about that later, is $150, second place $250, and then first place is $500, and also it's the opportunity to present representing the University of Minnesota at the Midwest Association of Graduate Schools meeting, which is a competition next spring. So with that, let me introduce our judges. And what I'd like is, I'm just going to advance the slides and have each judge introduce themselves, also share what inspired you to participate in this competition beyond Nuru twisting your arm. Pretty sure that was the main thing. And reflect briefly on the importance of strong communication skills. With that, Jade.
All right. Woo, this is loud, or I'm loud, can't tell. My name is Jade Denson. I lead a strategy and operations team within Target. I was inspired to say yes to Judge because although I work in corporate, I'm a nerd. I love learning. And to me, this was an opportunity to get back into learning something that I may not have known about. As far as communication and the value of that, so within my team at Target, I hope to drive strategy within the tech organization. And I like to say your idea is only as good as your ability to communicate it. You have to get people bought in. That's the heart of having a good idea too. And so within my work, helping to drive and implement strategy for tech, I have to convince people, I know what I'm talking about. I have to convince people that there's risk to not going this direction. And so strong communication is super helpful for that. Happy to be here.
Great, thank you.
I'm Chris Farrell. I'm Senior Economics Contributor at Minnesota Public Radio and Marketplace. And I was just really curious about the presentations and what are the things that people are working on. And I'm always just amazed just at the creativity of what people are exploring at the university. And of course, I'm in journalism, so storytelling matters. But the thing is, you're working in a story, you do all this work, you do all this research, and it turns out if you don't frame a story well, no one really pays any attention to what you did, even if what you said is really good. I mean, it's important information. So storytelling matters, how you frame a story matters, and how you reach out to your audience, doesn't matter what you're doing, is incredibly important.
Brilliant. Yes, Barb.
Good morning, and congratulations for being here, and thanks for sharing your stories with us. My career has been in technology and cybersecurity, so pretty technical. And my one thought I want to share with you is this, which is that your communication skills, in my experience in a technical field, your communication skills are what set the ceiling for your career, not your technical knowledge. If you think about as you're advancing your career and you're moving up and you're in conversations with your peers, at some point the people around the table will not know what you know. They don't understand your field. They're going to depend on you to crystallize your message and convey it in a way they can understand it so they in turn can make well-informed decisions. It may be decisions involving the direction of the organization, maybe decisions related to funding your work. So your ability to convey that message is really what determines your effectiveness in that role. So that's the reason I wanted to come, because I believe that strongly, and I'm super excited to hear what you have to share with us today. Best of luck today.
Good morning. I'm Rickette CSR. I'm the executive director for a local publishing arts initiative. And so kind of like Chris, the way in which you represent your idea and express it and convey the knowledge or whatever the content is of what you're doing is really important. As a school psychologist, a editor, a publisher, I come across a lot of great ideas and a lot of great stories, but like you said, I mean, there's some that just doesn't convey a human touch, or it might not convey the idea that you're trying to express. So stay strong to your idea, but also work on your art in terms of, and craft, in terms of how you convey it. So I'm happy to be here. This was a curiosity request or response. So thank you for being here, and I am really honored.
Good morning, everyone. My name is Brianna Smith. I'm the program manager for the Osher Lifelong Learning Institute here at the University of Minnesota. We call ourselves OLLI. I'm here because I think 3MT and OLLI are kindred spirits, in that, like 3MT, OLLI is really committed to helping our graduate students learn how to present their research to a non-specialist audience. In our OLLI Scholar classes, we invite our grad students and postdocs to teach 7-week courses for our OLLI members. Our OLLI members being inquisitive, active, older adult learners, average age 75. So whether in a 3-minute thesis or a 7-week OLLI course, I think this is a wonderful opportunity for our students to gain clarity about what they're doing its larger importance and to kind of tap into perhaps larger societal resonances of their work, which is applicable in many areas. So I'm so excited to join everyone today and really excited to get to the presentations.
Thank you. And thank you, judges, for taking time out of this, what I'm sure are very, very busy schedules, to help us with this. So today, we're honored to have with us Executive Vice President and Provost Gretchen Ritter, whose leadership and presence here today reflect her dedication to advancing academic excellence and supporting graduate student success at the University of Minnesota. Provost Ritter is the Chief Academic Officer of the University of Minnesota's 5 campuses and the Provost of the Twin Cities campus. She joins the university from Syracuse University, where she was the inaugural Vice President for Civic Engagement and Education. In this role, Ritter collaborated with academic programs and units to advance appreciation for civic engagement and community service while facilitating campus-wide programming to foster civil dialogue and free expression across viewpoints. During this time, she was a professor of political science. She just doesn't have enough to fill her days, so she's got to do all these other things as well. She was professor of political science in the Maxwell School of Citizenship and Public Affairs and a faculty affiliate with the Program for Advancement of Research on Conflict and Collaboration. Dr. Ritter previously served as the Vice Chancellor, Provost, and Chief Academic Officer at Syracuse University. Prior to that, she was the Executive Dean and Vice Provost of the College of Arts and Sciences at The Ohio State University. You'll have to explain that to me at some point, where that's from, but that's okay. And the Vice Provost of Undergraduate Education and Faculty Governance at the University of Texas at Austin. She holds a bachelor's degree in government from Cornell University and a doctorate in political science from Massachusetts Institute of Technology. Please join me in welcoming Provost Gretchen Ritter.
So, good morning, everyone. Oh, what did I do? No?
I don't know. That's weird.
Okay.
That's okay. You do your thing.
All right.
I'll come back.
We'll forget about that. So welcome to all of you to the 3-minute thesis competition. I'm really excited to hear our presentations. And thank you so much to our panel of judges. Great group. I'm excited for your participation in this event as well. Congratulations to our competitors from the 12 colleges across the university. And to our contestants, as you have likely discovered, the ability to sum up your research concisely and without jargon is a really valuable tool, and you've just heard that affirmed here. Over the course of your career, you'll have a lot of opportunities to share your research with your peers, but it's the ability to describe your work in ways that captures the interest and understanding of a lay audience. That's the next level skill. More broadly, learning to communicate clearly, keeping in mind your audience and the context, will benefit you no matter what career path you end up taking. Someday, you may end up pitching your ideas to a foundation funder. And being clear and concise about your work and why it matters could make all the difference. Or maybe you'll have a chance to talk to a legislator about your work on climate change and win their support for what you're trying to do. Or perhaps you'll end up taking a different path. Maybe you'll end up joining a company or a nonprofit and talking to them about why you believe that the arts, for instance, enhances productivity and well-being. That could increase the impact of the things that you care about. So being diligent and creative as a researcher is, of course, critical to your success. But so, too, in your career and in your life, will be being a good communicator. I want to congratulate all of you on having gotten as far as you have and on your commitment to strengthening your skills in this area. And the other thing I'll mention for you and any other graduate students who we have here, or future graduate students who we have here, is that there may be other skills that you'd like to strengthen as well. Skills in teaching, in grant writing, in team building, in management, or in mentoring. And the graduate school offers a wide range of professional development opportunities and resources. So I encourage all of you to spend a little time taking a look at their website and exploring those opportunities. So once again, congratulations. We're really proud of what you've already accomplished, and we look forward to your presentations.
All right, let's see if Scott can fix the technology. Ooh, wow. Thank you. All right, so before we get to our actual presentations, I want you to think about this People's Choice Awards as you go in and start listening to this. Each of you is going to have a chance to vote for your favorite presenter after all the talks have been concluded. And while I know that many of you are here to cheer for a particular person, and that's great, we do encourage you to use the judging criteria and cast your vote based on the presentation quality and communication skill. So it's time to begin. Please join me in welcoming our first presenter, Matthew Jacob from the chemistry program in the Swenson College of Science and Engineering on the Duluth campus.
Sorry.
Okay, this one.
She looked at her mom and asked, "Mom, could you please shave my head one last time before my chemotherapy starts?" Chemotherapy knocks you down before it knocks the cancer out. So here's the question. Can we make the treatment more effective and smarter with greater selectivity and fewer side effects? So I thought, what if I instead target the cancer cell's favorite energy source? The mitochondria, the powerhouse of the cell. Yes, the high school biology fact you thought you'll never use again, right? Turns out I'm using it now. Let's picture this. You take a cheap decades-old pill to get rid of worms, and it actually might fight cancer. Sounds like a plot twist in a medical drama, right? That's my research. The drug is called niclosamide. It's been around since our grandparents were kids. In the lab, it fights cancer cells effectively. But inside the body, it's like being stuck in the traffic. Most of the drug just doesn't get absorbed. So my project is about providing niclosamide, a flying cape, turning it into a super drug so that it dives straight into the cancer tumor target. But how? With our chemistry magic, or as researchers, we follow a process called salt formation. On its own, niclosamide is shy, it doesn't dissolve, and it won't mingle like the people who leave the party early. But if you pair it up with the right partner, boom, it suddenly becomes social, which is like providing niclosamide the VIP passport to get into the bloodstream. As part of this project, I have synthesized multiple different variations of the salt forms and tested its potency against 4 different cancer cell lines, and the results were genuinely exciting. Remember, this testing was done outside the body. Riding on that momentum, I moved inside the body. We injected our compound into mice to see how well they handle it. They were chill, no side effects, ate snacks, took naps, and lived their best moist life with no drama. Unlike my life. Now you might ask, why does this matter? Well, making a brand new drug costs billions of dollars and takes years. But if you repurpose a cheap existing drug, just by tweaking its form, could bring patients a faster and more affordable treatment. If you remember in the start, the daughter asked, "Mom, could you please shave my head one last time?" My research answers that be smarter and selective by giving an old drug a new life, so that heads are shared by choice, like Vin Diesel, not by chemo. Thank you.
I didn't explain, there'll be a pause between each while the judges do their work. So, thank you. Our second presenter is Vanessa Segura Duque from the Human Factors and Ergonomics program in the College of Design.
Imagine a child waiting weeks for a medical device that doesn't even fit, because most of these devices aren't truly designed for children. They are just smaller versions of adult models. And a child's face undergoes dramatic transformations in size, shape, and proportion from birth through adolescence. Static devices can't keep up with that growth. But during the first weeks, but every single week of waiting matters. When a child receives a medical device that doesn't fit, their therapy becomes less effective. And that means longer stays at the hospital, higher costs, and more stress, not just for the child and the family, but for the entire healthcare system. And that's why personalized medical devices offer a new way forward. Yet, in many hospitals, personalization is seen as something futuristic or unattainable. My work begins with a simple belief. Personalization shouldn't be a privilege. It should be a right. Every single kid deserves a device made for them, not to fit around them. Over the past years, I've studied dozens of attempts to create personalized medical devices. All of these are brilliant ideas, but isolated. Each one focuses on one device, one material, one patient. My research connects them, and I'm building a pipeline to design and produce personalized medical devices in just 24 hours. It starts with a quick contactless 3D scan of the child's face. Then this data fits into a parametric system where the 3D geometry guides and evaluates the shape of the device. Then, within hours, a 3D printer produces a soft, medical-grade silicone device ready for fitting. Amazing, isn't it? What used to take weeks now happens in a single day while the child is still in care. By developing this framework, this process can adapt to other devices. It could be a CPAP mask for sleep apnea, a nostril returner for cleft palate, and an ear stand for ear therapy. It turns custom design into a system hospitals can actually use. My dream is for this framework to live inside hospitals everywhere. Imagine a lab on the pediatric floor where designers and doctors work side by side. That's the future I imagine, a place where data meets empathy, where design meets medicine, and where no child has to wait weeks for a medical device that doesn't fit. Thank you.
Up next is Mary Rachel Danielle from the Experimental and Clinical Pharmacology in the College of Pharmacy.
Let me start with a story. Jack, a 10-year-old boy, won the battle against cancer. The drug that had saved his life, doxorubicin or Dox, is a cancer-fighting superhero. It swoops in, kills tumors, and saves lives. But like every superhero movie, there's a twist. Dox has a dangerous sidekick. It can quietly damage the heart, not only right away, but years later. And if kids like Jack grow up and develop hypertension or high blood pressure, that hidden damage could come back with a vengeance. In fact, childhood cancer survivors treated with Dox are 12 times more likely to develop heart failure if they also have hypertension. While most scientists have been focused directly towards the heart, the obvious battlefield, I decided to check what was happening off screen, the body's immune headquarters, the spleen. So here's my idea. The Dox doesn't just hurt the heart. It disrupts the immune soldiers in the spleen, particularly the inflammatory immune cells. Upon treatment with Dox, these cells start receiving faulty instructions. They wait quietly, but when hypertension is struck, the spleen opens its gates and sends those immune soldiers towards the heart, where they could cause more harm than help. To test this, I developed a unique two-hit mouse model that reflected Jack's clinical experience. I treated young mice with 3 rounds of Dox, similar to children in cancer therapy, and I let them grow up. Once they reached adulthood, I struck them with hypertension and watched what happened. The results were striking. The mice that were treated with Dox alone, their hearts seemed to handle the treatment relatively well. But when hypertension struck, everything changed. Their heart function dropped, inflammation flared, and those confused soldiers marched from spleen to the heart, causing more harm. When I repeated the experiment in mice that were lacking these inflammatory immune cells, it was like cutting off the bad radio signals. Those soldiers never got the wrong orders, and their hearts stayed protected. That's when the story came together. The heart-spleen axis is real, and it matters. So now my mission is to stop this faulty communication before it causes more harm. I'm exploring ways to stop these inflammatory immune cells from turning a rescue mission into a friendly fire. Because in the end, this research isn't just about mice or molecules. It's about kids like Jack. It's about making sure after winning the battle against cancer, their hearts stay protected for the lives they fought for. Thank you.
Our next presenter is Tobias Edwards from the Psychology Program, the College of Liberal Arts.
It was in 1990 that researchers analyzed twins who had been separated at birth. 35 pairs were identical, sharing all their DNA, and another 35 pairs were fraternal, sharing half their DNA. When the scientists reunited these twins, they made an incredible discovery. The genetically identical twins not only shared more genes, but they also shared more religious beliefs. The scientists learned that the importance you place in religion is partly genetic. This research was done right here at the University of Minnesota. 35 years on, we still do not know why religion is genetic, which genes are involved, and how do they function? My dissertation investigates this mystery by linking specific genes to religious faith. To identify the effects of genes requires a huge sample. So I partnered with several research institutions to amass 650,000 volunteers across dozens of countries. Each participant gave a blood or saliva sample and filled in a religion questionnaire. We then used their sample to sequence their DNA. So, with the religious values and DNA of 650,000 people, what did I find? I was able to identify hundreds of genes associated with religion. These genes had unusual properties. They were more likely to be expressed in the frontal lobe and prefrontal cortex, areas of the brain involved in planning, decision-making, and navigating social life. The genes were also involved in neurotransmission, the relay race of electrical signals from one neuron to the next. The most influential gene I discovered, the ZIP8 gene, is a well-known risk factor for schizophrenia, supporting an old theory that some religious experiences might be mediated by hallucinations. But what does all this biology tell us about your religious beliefs? For me, the real challenge is prediction. Is it actually possible to predict your religious beliefs from your DNA? To test this, I returned to the Minnesota twins. Now there are many of them, and they've all been genetically sequenced. From their DNA, I was able to predict a range of religious behaviors. How often they go to church, how often they read scripture, how frequently they pray, and even how spiritual they claim to be. Your relationship with God is wondrous, personal, and mysterious. Indeed, traces of it are written into your DNA. Thank you.
Next up is Lauren Kress from the dual PhD DDS program in oral biology from the School of Dentistry.
I hope you're all able to enjoy the beautiful fall weather that we're having here in Minnesota today. As Minnesotans, we know that this is probably not going to be long-lived as winter is rapidly approaching. Although we might not be looking forward to shoveling our driveways again or braving our snowy commutes, I think we can all take a huge collective sigh of relief that the cold weather means the end of our dreaded construction season. So as I was driving around the Twin Cities earlier this fall and noticing all of the new buildings, houses, and bridges that were going up, I was really struck by this connection that it seems to exist between construction and dental plaque formation. So you may be asking yourselves, what is this connection? So just like any building requires a firm foundation on which to construct the complex framework of the structure itself, dental plaque also requires a firm foundation. But rather than a concrete foundation, it requires a bacterial foundation. So today I'm going to be talking to you about one of these foundational bacterial species that I study called Streptococcus gordonii. So Streptococcus gordonii, as indicated here on my slide by the chain of green circles, is one of the very first bacterial species to come and bind to the tooth surface. And so once this bacterial foundation is laid by Streptococcus gordonii, other bacteria are able to come in and bind and interact with each other to form this complex layer of dental plaque that we can both see and sometimes feel on our tooth surface. So, although we know that Streptococcus gordonii is really important for building this foundation of dental plaque, there's still a lot we don't understand about how it knows when to start building this foundation. So that's where my research comes in. I'm specifically studying biological sensors that exist within this bacteria that allow it to sense not only internal but external environmental signals to trigger it to start building this foundation. So why is this important? Well, by understanding some of these mechanisms in this bacteria, we can maybe come up with new novel interventions to prevent dental plaque formation altogether on the tooth surface. Plus, since Streptococcus gordonii is very closely related to another species of bacteria that we know to cause cavities or tooth decay, my work also has important implications for preventing oral diseases. So, just like any building requires a firm foundation without which it would completely crumble, maybe by targeting the bacterial foundation of dental plaque or Streptococcus gordonii, we could prevent this building of the dental plaque on the tooth surface and also alleviate oral disease. Thank you so much.
Our next presenter is Jason Hatfield from the Comparative and Molecular Biosciences Department in the College of Veterinary Medicine.
A poultry farmer washes his hands and changes out his boots and overalls in a clean room, standard biosafety protocol, before walking into one of the many identical turkey coops holding 10,000 turkeys that his family's had for 3 generations. He brings in some feed, checks over his flock, and then leaves. Within 4 days, every one of those birds will be either dead or dying, and he'll be forced to cull the birds in order to save the rest of his farm. This very scene is playing out over and over and over again in the state of Minnesota and across the U.S. as farmers are being faced with one of the worst avian respiratory disease outbreaks in nearly a decade, all caused by a virus that most people aren't even aware exists. Avian metapneumovirus is a respiratory disease that has a major impact on the poultry industry. In just the last 8 months, it's cost over $112 million to the state of Minnesota alone, with some estimates as high as $1 billion nationwide. Now, it is important to note that the virus in and of itself doesn't cause high mortality. However, it is extremely contagious and uniquely able to cripple the immune system of young animals, often leading to dangerous and deadly widespread secondary infections. In older birds, it decreases egg production and weight gain, affecting the entire industry. Now, how this virus is spreading into these biosecure facilities, and more importantly, how to prevent it, is the literal billion-dollar question I'm trying to answer. I test how long this virus can survive on common materials found in poultry barns like plastic, cardboard, and metal. I see how different factors like temperature and humidity can affect the survivability of the virus and how easily it's able to become airborne and spread. But my work goes beyond just seeing what this virus does. I'm also actively trying to stop it from spreading using a form of ultraviolet light called far UVC light. Now, unlike traditional UV light, which can cause sunburns and skin cancer, far UVC light operates at a very specific lower wavelength. What that means is that it's significantly safer for both human and animal exposure, as it cannot harm our skin, yet it can still kill pathogens. In my lab, I test whether avian metapneumovirus can be inactivated by far UVC light on surfaces, in the air, and perhaps most importantly of all, how we can actually apply this to real-world settings. If successful, this research could lead to better disinfection strategies, not only for avian metapneumovirus, but other airborne diseases as well, found in both animals and humans, like COVID, influenza, or even the common cold. That's because this is a One Health approach. Because when we protect animal health, we're also protecting human health. In an increasingly globalized world where viruses move more freely than ever before, we need tools that can operate across all different areas of life. And sometimes that tool is as simple and as powerful as a safe, invisible light. Thank you.
Next, please welcome Sarah Zeiman from the Civil Engineering Program in the College of Science and Engineering.
Katy Perry once said, "Do you ever feel like a plastic bag drifting through the wind, breaking into microplastics. Okay, maybe she didn't say that last part, but I bet your ears perked up at the term microplastics. There's a growing public health concern about how toxic and how widespread these particles are. But it can be difficult and labor-intensive to measure the size, type, and presence of microplastics in all of our ecosystems. Nanoplastics, which are even smaller than microplastics, pose more serious problems, as they can be ingested more easily and they are more challenging to detect. My research focuses on how micro and nanoplastics are generated from the breakdown of our large plastic products, which is necessary to understand how much plastic we are being exposed to and how toxic it could potentially be. I am taking a data-driven approach to compile nearly 300 studies on plastic degradation in the natural environment. My analysis can inform a risk assessment, which can help us to mitigate our colossal plastic waste problem. See, the thing is, unlike other pollutants, plastic degrades in a multi-step and multi-phase reaction. So there are different types of measurements to study a plastic's breakdown. So far, I found that both the type of plastic and the environment that it's in can be important predictors in how and how quickly a plastic will degrade into further products. This can help us to understand how, why, when, and where micro and nanoplastics exist in nature, which then can allow us to make smarter choices initially about the plastics that we use. As of today, there is a lack of data and understanding in specifically how mechanical forces degrade plastic in the environment. For my next step, I address this gap by focusing on one mechanical degradation mechanism called abrasive wear. This happens when hard sand slides against plastic, which occurs widely in beaches, riverbeds, and desert environments. We simulated these interactions in the lab by creating a custom sand abrasion device. Surprisingly, we found that the plastic film strictly released nano-sized particles, and there were nanoplastics stuck to the sand. In the environment, the plastic-coated sand could be washed downstream or blown by the wind, and further distribute these nanoplastics in new environments. This plastic-coated sand could also interact with other known harmful pollutants, like heavy metals or pesticides. My research on a plastic bag drifting through the wind may not be sung to a catchy tune like the Katy Perry song, but it will help us to better understand how our plastic products are changing human health and impacting the environment. Thank you.
Our next presenter is Nisha Vishwanathan from the Biochemistry, Molecular Biology, and Biophysics graduate program in the College of Biological Sciences.
All right, how am I feeling today? Alive? Healthy? The truth is, many of us in this room are here today because antibiotics once saved our lives, or the lives of our parents, or someone we love. But now we are on the edge of a global crisis because bacteria are fighting back. Scientists recently warned us about the rise of nightmare bacteria. These are germs that have become so dangerously resistant that even the best of our antibiotics cannot kill them. It's a true nightmare to doctors and to all of us because suddenly a simple cut, a scratch, or a sore throat could become a death sentence. It's a silent pandemic we are walking into. And that's why we urgently need better weapons. But here's the twist. Many of our best antibiotics were inspired by bacteria themselves. If you zoom closely into the microscopic world, there's a constant war between germs. A real-life Game of Thrones with endless battle for territory and for survival. To compete, bacteria produce these remarkable antibiotic weapons, molecules that can kill, disarm, or outsmart their neighbors. And that's where my research comes in. Our lab has discovered a set of these natural molecules with a built-in armor. You can imagine them as these tiny soldiers. You see nature's armored drugs behind that shield on that screen. Each one is protected by a special chemical modification, those little CH3s you see on their backbone. This critical feature acts like an armor plating, transforming these ordinary soldiers into stronger, tougher, and a more resilient antibiotic army. So why does this matter to us, and how does it work? Normally, when you take a drug, our body often tries to break it down before it can take full effect. This natural armor can protect the drug, giving it the time to stay active and effective. And chemists struggle to design these kind of armored drugs, but certain bacteria do it so gracefully. They are the masters at installing this unique design. So by studying bacteria's DNA and the molecular machinery that craft these armored drugs, I aim at leveraging nature's own design to develop tomorrow's stronger medicine, helping us stay one step ahead in this ongoing battle against nightmare bacteria. So stay safe, healthy, and happy. Thank you.
Next up is Chani Kim from nursing program, PhD program in the School of Nursing.
Did you know that measles, a disease once declared eliminated in the US, has now been reported in nearly 1,700 cases this year alone? But have you ever wondered, how do we even know that these outbreaks are happening? How do we detect the moment when a few isolated infections turn into public health emergency? The answer lies in something like a fire alarm, a system designed to detect danger before it spreads. In public health, that fire alarm is called disease surveillance. Think of when you cook a steak. A bit of smoke rises, nothing alarming yet. But if you burn it, the smoke thickens, and the fire alarm goes off to stop a real fire from spreading. Disease surveillance works the same way. There are always some cases in every community, but when infections start to increase rapidly, our public health fire alarm must respond quickly. Traditionally, when clinicians, for example, nurses like me, identify a case of infectious disease, they have to check all the required information and fill out a report and send it to public health agency by fax, mail, or phone call. The whole process relied on human decision and manual work. It's like having a fire alarm that only works when someone remembers to push the button. I'm studying a system called ECR, electronic case reporting, which is like an automatic fire alarm for infectious diseases. As soon as diagnosis code or positive lab test is entered into patient health record, that information immediately triggers the ECR system to generate a report and send it directly to the public health agency. It's automatic fire alarm for infectious diseases. The alarm sounds the moment the first smoke appears. My research focuses on evaluating how effective the ECR really is and how it can be improved. I analyzed surveillance data from Minnesota Department of Health to assess how timely and complete the ECR data is. I also conduct several one-hour interviews with ECR users to understand their challenges and find a way to improve the system. By combining both of these insights, this study aims to make ECR faster, more accurate, and easier to use. Because the earlier we detect, the sooner we can respond and save more lives. I believe this research helps us keep our attention not on the paperwork or computer screen, but on the people and community we aim to protect. The next time you hear an outbreak has been detected, remember that somewhere a quiet electronic fire alarm has already gone off. Thank you.
Our next presenter is Alina Smolskaya from the Applied Plant Sciences program in the College of Food, Agricultural, and Natural Resource Sciences.
When is the last time you ate soybean? Perhaps it was when you had soy sauce with your sushi, or did edamame as an appetizer. But what if I told you that if you've eaten any animal products or cooked anything in vegetable oil, you've likely been touched by soybean as well? That's because most soy that leaves a farmer's field goes into the production of animal feed and oil. Today, there's a major challenge facing not just soybean farmers, but all of agriculture. It's estimated that by 2050, we need to more than double our current food production in order to keep up with population growth. As a soybean geneticist, my role in this challenge is to develop new, high-yielding soybean varieties for farmers so they can produce more on the same amount of land. Soybean yield is made up of how many seeds the farmer harvests. It's a function of how many plants they have in their field, how many pods are on those plants, and how many seeds are inside each pod. I research the number of seeds that the plant makes. More specifically, my thesis aims to answer the following question. What gene controls how many seeds are in a pod? If we know this gene, we can use that knowledge to develop new, high-yielding soybean varieties. So how do you find a gene? To find the gene controlling seed number, I started out by playing soybean matchmaker. I took two plants that were different for how many seeds they made in their pods. One plant produced few seeds, while the other produced many. I then took those two plants and cross-pollinated them, so they made hundreds of offspring. Just as you inherit genes for eye color from your parents' DNA, these plants are inheriting genes for seed number. And looking through the offspring, I saw that some produced lots of seeds, while others produced few, indicating that they have a difference in how many seeds they make in the gene that controls how many seeds they make. So looking at our example, I separated those plants into two groups, those with low seed production and those with high seed production. And then I look for differences between the two groups at the DNA level. In our example, you can see the plants and their DNA represented below them. And for simplicity, let's imagine each plant only has 3 genes. Looking at the two groups, we see that the second gene is always different, indicating that this is the gene controlling seed number. Currently in my research, I'm in this step, looking at the DNA of the different plants. If we find the gene that controls seed number, we can use that knowledge to develop new, high-yielding soybean varieties for our farmers so they can produce more with less for people like me and you.
Up next is Itorobong Uwat from the Environmental Health Sciences in the School of Public Health.
If you watched Alice in Wonderland as a kid, did you ever wonder what this caterpillar was puffing? To me, it just seemed whimsical because I didn't know what it was back then. It wasn't until recently, when the scene flashed back in my mind, that I realized that's a hookah, which is also known as a water pipe. This 1950s classic portrays that it was okay to smoke hookah around a kid then. Now here we are in 2025, and that perception hasn't changed in reality. This photo still mirrors current scenarios, like a dear friend of mine lounging with a hookah at home with kids nearby. Young adults in the US enjoying hookah around younger siblings, or even immigrant families gathering with hookah as a centerpiece. Beyond the potential exposure in kids, what else do these scenarios share in common? It's the misconception that hookah is safe, which is fueled by a lack of awareness, policy gaps, and the components of hookah itself, like its appealing flavors, the sugars that mask the harshness of the smoke, and the water that many believe purifies it, all feed this false sense of safety. What if I told you that a hookah session lasting 45 to 90 minutes compared to just 3 to 5 minutes of a cigarette generates about 27 times more of a human carcinogen called formaldehyde and 3 times more addictive nicotine? Surprising, right? Well, here is the kicker. Despite this, hookah tobacco is exempt from most tobacco control policies. It's like the mischievous sibling of the tobacco family that somehow gets away with anything. So when I say I'm on a mission to demystify the sweet deception surrounding hookah, you would understand why. My research investigates how the flavors, sugars, and toxicants in hookah tobacco influence smoking behaviors and risk perceptions. I'll examine how these factors increase the risk of exposure in kids living with users through air sampling, chemical profiling, and survey evaluations. Speaking of kids, these kids are exposed without choice, but yet more vulnerable due to their faster breeding rates and immature immunity. So I'd also work with high prevalence communities to raise awareness on secondhand smoke exposure and advocate for changes in smoking behaviors. Because the bitter truth is, behind the appeal of hookah tobacco lie health consequences that affect more than just the users, but loved ones that we wish to protect. Thank you.
Our final talk this morning is Olivia Kukan from the Molecular Pharmacology and Therapeutics program in the medical school.
Wow. You can tell that the construction was bad last year when 2 competitors decided to pull the construction analogy. Today I'm going to talk to you about my thesis research in terms of the 2 construction sites behind me, and my thesis research is in breast cancer immunology. So breast cancer is a disease that has surely somehow affected the lives of nearly every single person in this room. And while we're getting better at treating breast cancer, some patients still don't respond well to our current therapies. So understanding the science of breast cancer and how our bodies respond to it is a crucial next step in the discovery of better treatments. If you look at the first construction site behind me, we see the normal wound healing process. And I study a special type of immune cells called macrophages. So when you get a paper cut, you open up your skin to germs, bacteria, and debris. And what macrophages do is they come in, identify the germs, eat the germs, and alert the other cells in the immune system. Now, eventually, the paper cut does close and begin to heal. And the really cool thing about macrophages is that they're involved with that process too, rebuilding the skin tissue, helping scab, and telling the inflammatory immune system, we don't need you, we've got it handled. We step over To the second construction site, we find the breast tumor microenvironment, which consists not only of the cancer cells, but also the immune cells and other components within the tumor. And these orderly steps of macrophages in that wound healing process become really disorganized and can even be used by the tumor to help support its growth. And so just like a construction worker has the capability to be both demolishing and building, taking a jackhammer to the road or laying down new asphalt depending on the orders it's being given, macrophages can also be demolishing or building depending on the cues that they're getting. So a demolishing macrophage sees the tumor as that early paper cut, alerting the other cells in the immune system and killing the tumor cells as it would to germs in the cut. However, a building macrophage sees the tumor more as that scabbing wound, suppressing the inflammatory immune system and building structural support that helps the tumor grow and spread to other parts of the body. So my thesis has two big goals. The first is to understand what cues in the tumor microenvironment are driving these demolishing and building responses. And the second is to use engineering to give molecular instructions to these immune cells to be more pro-demolishing, to kill the tumor cells and take a wrecking ball to the tumor, ultimately leading to better outcomes for breast cancer patients and their families. Thank you.
I think our judges have written more in the last 40 minutes than they have for the last week, maybe. So our judges will now confer to determine the winners, and it's time for you to cast your vote for the People's Choice Award. But before you do, can we have a big round of applause for our presenters? So if you take out your handy electronic device and scan this QR code, you'll have a chance to cast your vote for the People's Choice Award. Oh, good. We're checking on that. I have people who are doing that, because I don't know.
If you need paper,
Yeah, let's do that. Old school, we're going paper. Nuru says hers worked, so keep trying until you get a paper ballot. And I shouldn't have to say this, but if you already voted on the poll, don't take a paper ballot. So, if you've already voted, we have a reception area outside of the theater, some refreshment there. I encourage you to go out. Take about 20 minutes before the judges will be done. They're nodding their heads. Good luck with that 20 minutes. But please feel free to go out into the lobby as soon as you've voted and enjoy the refreshments.
Thank you.
Yep.
I'll get them.
I think maybe I might leave the QR code up just a little bit longer, just in case.
People need it. Okay.
Yeah. And we want them to go out anyway.
Yes.
So, as always, oh my God.
It's pretty awesome.
Almost there, almost there. We gotta get everybody back in here. That's it.
So,
First, we're gonna do
I'm gonna
the.
call them up.
Yep.
Great. The silver one is already
Okay.
Okay.
So you're gonna call them up and ask them to stand in the order that they presented. So the first one will stand next to you.
Okay.
So forth. Yep.
We're doing giant check.
Oh yeah, giant check.
Are we writing it up?
Yes.
To Ito?
Yes.
How much?
500. Yeah. Thank you.
Okay, so I'll hand these to them.
Yes.
Okay.
Is that
You
cool?
can just put them down here.
Okay. And then we'll do People's Choice first.
Yep.
Then the second, and then.
Okay.
All right, we gotta get people back in here though.
All righty. Phone. Missing
Oh.
phone.
Okay. Okay.
Molly says to appear to Kevin to turn the music off. I don't know about the check. I haven't seen that yet. I heard them talk about it. No. All right. Thank you all. Thank you all for voting for People's Choice. I'm going to have the finalists come up here and stand on stage. Matthew, if you could be on this end and do the order that you were speaking in, out to the side here. And this is a good time to remind you all. So you've just seen 12 amazing presentations summarizing the research very quickly that these students are doing here at the University of Minnesota. We have 7,000 graduate students here at the university. Keep that in mind as you think about this. So I have certificates for you first. Drum roll. You only get to touch it for a few seconds. All right. And next, our second place. Photographers control the world. And our first place goes to, and again, there's a monetary prize with this, but also the opportunity to compete at the Midwest Association of Graduate Schools competition next spring. Again, I want to thank everybody here for coming. I want to thank all the mentors for mentoring these students, and really just remember, this is what a research university does. We help really sort of fulfill these dreams, help these students fulfill their dreams for the research that they want to do to change society. And you heard that today. So thank you all. Have a great day. You can leave the stage. Oh, I'm sorry. Okay.
I must have grabbed something when I was talking. It's green.
Someone was missing a phone. Someone lost a phone. Okay. I think Nuru just went to get them. Sorry.
Just jump forward for 1 sec.
Oops, that was me. That was me. Running to the next meeting. Thank you very much. Good job. Sure. Oh, with her slide. Yep. There you go.